Good morning to all colleagues who have joined. Friends, as you know, this is a very important lecture series by eminent persons, eminent speakers. And the important thing is that it's a very important in the sense that it's being organized to celebrate a very proud moment for every Indian, that is to celebrate the completion of 75 years of India's independence. We are all proud of our great country. And to celebrate this, the ICAR, Indian Council of Agricultural Research, has taken an initiative to conduct 75 lecture series, in addition to many other activities to celebrate this great moment. Many other important activities are being conducted by ICAR for the nutritional security and many more things. And in that series, we have completed 35 lectures till date. And today is the 36th lecture by a very prominent personality, Professor Chandrima Shaha. She doesn't require much introduction. She's a well-known figure. Dr. Chandrima Shaha is president of the Indian National Science Academy, a very prominent academy, and J.C. Bose Chair Professor at the Indian Institute of Chemical Biology. She is also the former Director and Professor of Eminence of the National Institute of Immunology at New Delhi. Her research interests center around the elucidation of the processes that influence cell death programs under varying physiological conditions in diverse organisms. She graduated from Calcutta University and did postdoctoral studies at the Kansas University Medical Center and Biomedical Center of the Population Council at the Rockefeller University, USA. She is an elected fellow of the World Academy of Sciences, all the three science academies of the India. She served as Vice President of International Affairs of the Indian National Science Academy and as a member in the councils of all three national academies. Notable awards include the Ranbaxy Science Foundation Award for Basic Sciences, the J.C. Board Fellowship, Shanti Swarup Bhatnagar Medal of INSA, Om Prakash Bhasin Award, Archana Sharma Memorial Award, Darshan Ranganathan Memorial Award, Chandrikala Pura Memorial Medal, and Shakuntala Amir Chand Prize. Many more awards she has won, and she's a well-known personality. We welcome you, madam, and we thank you very much for agreeing to deliver this important lecture on emerging disease and our immunity. I would like to inform you, madam, that this lecture is being live streamed. This lecture is also available on one of the customized website, and that is ICR75lectureseries.webconevents.com. And in this platform, the Zoom platform, we allow only the senior persons. The others, they are attending to those other two platforms. And in this platform, we have the many vice chancellors from the senior officers from ICR and the senior faculty from the universities also. Our Director General is just shortly joining, Dr. Mahapatra, but he asked me to start this lecture. Then our Deputy Director Generals are also here. So I once again thank you very much for accepting our request, and I request you to kindly take the floor and please give your lecture on this topic, emerging disease and our immunity. Thank you very much, madam.
Thank you, Dr. Agarwal, for the kind introduction. I would like to thank Dr. Mahapatra, yourself, and Dr. Singh, and others who are today here to for this lecture. And I'm most pleased to be in this forum and in the Azadi Ka Amrit Mahotsav celebrations. And today I chose a topic that that is interesting to people currently. And so I wanted to talk about emerging diseases, and there are so many questions about our immunity. So giving it, since it is a kind of lecture, so I have made it as understandable as possible for even students. So I would seek indulgence from experts in the field. And if I can share my slides now, yes. Yeah. Are my slides visible?
Yes, ma'am. Yes.
Yeah. Okay. So yeah. So, no, it's not changing. So I have to go back. It's not changing. Just shared. Are you seeing the? Sorry. I'm sorry. I don't know why.
Initially, when you shared, they were all coming before this lecture.
It's coming. It's coming, but it's not changing. It's changing now.
Yeah.
Okay. Sorry. Yeah. So the emerging first, if we consider what are emerging infections, we can see that new and emerging infections are those whose incidence in humans have increased within the past 2 decades, or whose incidence threatens to increase in the near future. Now, we know that infectious diseases have for centuries, ranked with wars and famine as major impediments to the progress and survival of humans. And they still remain among the leading causes of death and mortality in most part of the world. And with the COVID-19 coming in, we know that how infectious diseases can disrupt all our lives. So for emerging infections, it's like previously unknown infectious agents or pathogens which evolved over time. Even the SARS-CoV-2, the SARS-CoV at present, who caused epidemics beforehand. Whose role in specific diseases has previously gone unrecognized. We knew about known pathogens, but they're causing new symptoms, new diseases were not known. Those are also emerging pathogens. And known infectious agents that have spread to new geographic locations or new populations. And also re-emergence of pathogens, which we thought we have eliminated. And that is also an emerging disease. And known agents that have mutated to new forms. This is a very common occurrence where they mutate to new forms. And also, as you can see, this is a infographic from the Nature Medicine, that Zeonatic transmission has become a major problem because more and more the humans are coming in contact with animals who used to be in the jungles, who used to be outside the periphery of the cities, but more and more we are encroaching on their area. And we are getting all kinds of viruses that can jump species and cause havoc in humans. And that is what is suspected of the coronavirus, that it has come from bat. So now, what are the major contributors to the emerging infectious diseases? As I said, that human demographic changes because we have extended into areas where we're not supposed to extend, but that is in development and progress. Then behavior changes, human behavior changes, what we eat, how we deal with animals all around us, and how we deal with society also as a whole. Then changing industry practices, and also it changes in land use patterns. Land use pattern has widely changed. So that is one of the reasons. An increase in international travel and commerce is one of the means by which diseases can spread very easily. And after all these lockdowns, stopping of flights, you see COVID-19 sort of got controlled, but then again, it has started spreading along with the commencement of the international travel. And microbial adaptation to change is also another issue which helps to contribute towards emergent infectious diseases, because the microbial community keeps on changing their adaptation to a variety of environments. And also breakdown in healthcare system is another issue that can happen. And that has happened with the COVID-19 when it all of a sudden struck. There was a sort of breakdown in the healthcare system because it was simply overwhelmed. So this is one of the reasons that can happen. So if you look at this slide, which is a infographic from 2000, where you see the red ones are the newly emerging diseases like the Chandrima virus, the Bourbon virus, the Hunter virus. So these are the newly emerging diseases, and re-emerging diseases are denoted in blue, which includes like cholera, the Ebola, the diphtheria, Rift Valley fever, typhoid fever, all these. Then deliberately emerging is only one spot, which is the anthrax, which happened in the United States. We don't know if there are any other deliberately emerging diseases. So this is an infographic which was shared into September 2017, and already it has shown so many different diseases that can be categorized as emerging diseases. Now, if we go back to the germ theory, that the diseases are caused by germs was perceived as early as the Middle Ages, when a Persian polymath, Ibn Sina, who was one of the most significant physicians of the Islamic Golden Age, and he was a very famous physician and father of early modern medicine, he said the diseases were caused by impure substances. Fracastro, who was an Italian physician in the 16th century, he proposed that a scientific germ theory of disease more than 300 years before Louis Pasteur came in. And he sort of said that diseases were caused by seed-like entities. So in both these instances, you can see that they recognized that this was something external to the body that was coming in and causing the disease. But finally, in 18th century, Anthony van Leeuwenhoek, who was a Dutch draper who used to make lenses to look at his fiber, he discovered the microscope, which looked something like this. And this microscope is like it's like a small contraption where this is a very high power lens. And he first looked at what he called animalcules, moving bodies in a pond water or spermatozoa or blood cells. And he made sketches of the animalcules. And this is when actually it became evident that there was a world which was not what we cannot see with our naked eye. So then in the 19th century, two giants and Louis Pasteur brought about a lot of ideas in infectious diseases. The Charles Darwin's theory of natural selection, which he proposed in 1859, said that organisms most suited to their environment are the ones that can adjust to the prevailing environment and reproduce most successfully to pass on their traits to the next generation. And selective forces acting on the traits generates organisms best suited for survival. So his theory of natural selection, which actually picks that a trait can change depending on the environment and can be and the ones that can be transmitted to the next generation are the most suited for survival, for sustenance. Then Louis Pasteur discovered the theory of spontaneous generation. Actually disproved the theory of spontaneous generation, and he discovered that the bacteria came from previous bacteria and showed that after several passages in new hosts, the virulence of a microbial strain can increase. And by this, new virulence and new contagions can be created. So he was actually looking at selection in a way when he was doing these several passages, and natural selection acting on these pathogens were changing the pathogens. He proposed that evolutionary biology can help us in understanding of infectious diseases. The idea of harnessing ecology and evolution for controlling infectious diseases can therefore be traced back to these two stalwarts, Charles Darwin and Louis Pasteur. And even today, Charles Darwin's theory of natural selection applies to most of our understanding of infectious diseases. So if you compare the evolution of a bacteria and evolution of the humans, You see, the bacteria are the fastest reproducing organisms, doubling every 4 to 20 minutes. So they can undergo many mutations, whereas human evolution is a lengthy process of change by which people originated from ape-like ancestors. And the modern man came within 200,000 years. And from ape to man, it was about 6 million years. The out of Africa theory says that modern man walked out of Africa at about 70,000 years. Now, there were about 14, 15 species of man, which actually many of them disappeared. And so one species only survived. And the populations that happened out of Africa were extinguished with time. So, if you look at the generation time for bacteria is 12 minutes to 24 hours. And for humans is 22 to 33 years. So for us to adapt to the changing bacteria, it's a challenge for our immune system. And there are micro changes that do happen in the immune system, but we take much more time to evolve than a set of bacteria. So the race against pathogens, there is a race against pathogens that happens. And if you look at this, you'll see that Darwin's, there is a so selective forces are acting on the host to defeat an infection. Selective forces are acting on the pathogen to create an infection. So these two counter forces during evolution are actually acting on both the host and the pathogen. And this has over the years have shaped our immune system. So Darwin's concept of selection of the fittest, survival of the fittest, applies to the host who is best suited to thwart an infection survives, and a pathogen who are best suited to create an infection survives. So in this competition of the two, our immune system evolved, the pathogens themselves evolved, host also evolved many arsenal. So we are still surviving. So the proximity between the study of emergence and Darwin's views has increased with the advent of microbial phylogeny. We all know that genetic sequences from populations of microbes infecting one or several individuals are used to infer phylogenies. And these may contain key epidemiological information on the way infectious diseases spread, leaving footprints in their genomes as postulated by the field of phylodynamics. And it is important to understand the transforming epidemiology of the virus, as it helps in planning taking necessary steps for physicians and policymakers. Because if we can understand the epidemiology of a virus, if we can trace its origin, if we can trace its distribution, we can actually anticipate the possible turn of events that will occur with the pathogen. And then we can predict that when to hit. So this happens with mutations. Some mutations are harmful, while others can provide an advantage. And here is where Darwin's theory of natural selection comes in. And if a mutation gives the pathogen an advantage in a given environment in terms of survival and spread, this pathogen will survive better than its neighbors and its selected for. So mutations may alter many things. It can alter pathogen biology substantially, such as pathogenicity, infectivity, transmissibility, and antigenicity contributing to fitness advantage. Now, one of the examples that we can really think of is the bacteria, the drug-resistant bacteria. So in a population of drug-resistant bacteria, you should give antibiotics. Then only the drug-resistant ones will survive and propagate. So which shows that there is a selection taking place here under the pressure of antibiotics, similar to what happens in environmental pressure. So mutations actually can make a bacteria resistant to antibiotics, Some spontaneous mutations may make the bacterium resistant to antibiotic, and therefore in a given population, those that are able to survive in this environment will adapt through mutation and will survive. So this is how, we have to treat the infectious diseases as well, their changes. Now, when humans were hunter-gatherers and led more or less solitary lives, they were isolated and diseases did not spread that well. Then they gathered into small families, villages were formed, and eventually large communities were formed. So then the diseases, when large communities were formed, diseases, once it came in, started spreading within the community. It spread also to distant communities. So virus, bacteria, fungi, parasites, all kinds of diseases caused by these agents were spreading amongst the humans. Once the community, also at the same time, we have to realize that the population also increased. So eventually what happened is pandemics came in because the diseases started to spread. So the earliest evidence of smallpox actually dates to the 3rd century BCE, caused by the variola virus. And we all know that smallpox has been eliminated from the world. And that's why is that we don't understand really that why we were so successful in eliminating smallpox. One of the reasons may be that it doesn't have an animal reserve. And so in 1980, it was declared the smallpox is eradicated. Spanish flu, also known as the 1918 influenza pandemic, was caused by H1N1 virus. The most fatal pandemic was the plague. If you look at here, the Black Death or plague came in the 14th century. It also came in the 19th century. And the smallpox, which happened in about 8th century and 16th century onwards also, it got eliminated. But Black Death or plague also keeps on coming back in smaller forms. So 81 to present, we have the HIV, say it can be called a pandemic or an epidemic, but this HIV actually does not have a vaccine yet after 40 years of research. So this is an infographic from Nature Medicine, which came out very recently, and it shows the 21st century viral outbreaks. So the dengue, the SARS-CoV, which happened in 2002, then chikungunya, Rift Valley fever, H1N1 influenza, East Asia, Zika virus, Ebola virus, yellow fever, Nipah virus, then SARS-CoV-2 eventually in 2020. So we have been in the 21st century. We just crossed the first two decades, and we've been exposed to so many of these viruses. So it's a major problem for the healthcare system to take care of so many potentially coming diseases. So coming to this, RNA viruses, the reason I chose RNA viruses because that is the most interesting one for us and most relevant one for us now. These RNA viruses are, they typically have a spike glycoprotein. They have RNA and protein inside. Then they have the envelope, and there are some enzymes over here. Now, new genetic variants are constantly being created. One of the reasons is thought that because their RNA virus polymerase has no proofreading. The RNA virus is readily adapt to changing environmental conditions. Their ability to adapt is one reason why they survive so well. And the high mutation rate compared with DNA organisms for their enormous variability and adaptive capability. In case of COVID-19, the rate of evolution of SARS-CoV-2 from December 2019 to October 20 were about 2 mutations per month. So this mutations for these viruses are very interesting because if you look at this mutation at 614 position, which was from a D to it happened to a G, glycine, and aspartic acid to glycine, and the glycine helped the virus gave a selective advantage to the virus, and it transmits more readily than the 614D. And it was found that the number of functional spikes are more in the mutant, so which makes it better suited to latch on to the host cell. Patients infected with this G variant had higher viral loads in their upper airways than did patients with the D virus. So that higher viral load can lead to increased transmission when that particular patient actually coughs out the virus. So population genetic analysis indicates that the 614G increases in frequency relative to the 614D in a manner consistent with selective advantage. And if you look at this, this picture of England, where the 614G gradually, covered almost the whole of the country instead of the D, which disappeared slowly. So this is how this mutation was giving an advantage to the virus. And so there are other substitutions like this N439K. It also emerged and circulated widely in many European countries, and it was the asparaginase, the 439 site was replaced by lysine, and it enhanced the binding affinity for the ACE2 receptor. So most of the mutations occur in the region which it binds to the ACE2 receptor on the surface of the host cell, and this is the spike protein of the virus, and this is a three-dimensional structural drawing. So another mutation was 500 and N501Y mutation, where N went to Y, asparagine to tyrosine, and that also enhanced viral transmission seen with the combined mutation. So it suggests that it is a major determinant responsible for increased transmission of this variant. So this is how the mutations play a big role in survival of the virus. There is another thing which is adaptation to the new host. So chikungunya virus had an outbreak in 2005 in La Réunion Island, which is an island in the Indian Ocean under the French rule. And there a huge outbreak, a small outbreak occurred in 2005. But then in 2006, a huge outbreak came. So what was the reason? Because most of the viruses bore a key mutation in the position 226 of the E1 protein. And that increased the vectorial capacity of its host, which is Aedes albopictus mosquitoes. And their behavior is very anthropophilic, so that they bite more. And the Aedes aegypti, which was their usual vector, was bettered by the Aedes albopictus, which actually bit more people, were able to bite more people. So they changed a vector. The mutation in the virus actually helped the virus to change a vector and then spread itself. So this is an adaptation to the new host that also happens. Then something called evolutionary rescue, which is a process by which a potentially vulnerable population that could go extinct in the absence of evolution persists due to natural selection acting on heritable variations. So if you look at the resident strain, which is milder, causes milder disease, but remains around more time because it doesn't kill people. But it will peter off and disappear if it was like this. So the mutation comes in, and there is another peak, which gives a boost to this population, which of course the virus changes. But this is called a kind of evolutionary rescue, and it can persist still long enough. So this persists long enough for a mutation even to happen, so that it can survive through another set of new mutated pathogen. So there is another thing called trade-off, where evolution in which evolution is not able to optimize one part of a biological system without compromising another part of it. Now, highly virulent virus strains have counter selection because killing their host is detrimental to their epidemiological fitness. So any virus that is very virulent and kills its host is selected out, because then the virus cannot survive without the host. So HIV virulence is measured at the inverse of the time to get AIDS when no treatment is given. Without any treatment, eventually HIV will cause AIDS. So increased virulence for HIV comes in with increased probability of transmission per sexual contact, but the subject may have short survival. So the trade-off here originates from these two opposite forces. Milder viruses cause longer but poorly contiguous infections, and whereas virulent viruses cause short but contiguous infections. So this is a dilemma that selection that happens to selection, and one is given advantage over the other. So this is a very difficult situation, but this kind of trade-off is frequently seen. Now coming to the immune system, our immune system has two components. One is innate immunity and another is adaptive immunity. The first immune reaction was shown by Elie Mechnikov, who was a Russian scientist, and he that if he puts a thorn in a jellyfish, a rose thorn, there is an immune reaction. All the cells sort of home in here as if to throw off the thorn. So this, we have learned much about it now, and Mechnikov, who got the Nobel Prize in 1908, actually studied many of these immune cells, and he was a very famous contributor to immunology. So there are two types of cells. There is a third also, and there is a fourth also, but the two main types are dendritic cells and macrophages, who are the main players of innate immunity. And what they do is they first engulf the pathogen, try to eliminate it through their own mechanisms. And if they cannot, they call on the adaptive immunity, where they call on the help of, they actually chew up the antigens, present it on their surface, and they call on the B cell help and the T cell help. So when they present the antigens to the T cells will call on the B cells. So what happens is that one aspect is killing of the pathogen, and another is they produce interference also, and activation of T cells happen. T cells also generate interference, and they go and activate B cells. And these B cells differentiate into plasma cells and produce antibodies. Cytotoxic T elicited this is cytotoxic T cells killing a tumor cell, but the infected cells are also killed like this. So cytotoxic T cells can directly kill the pathogen. So there is an innate immune arm and the adaptive immune arm. So the innate immunity happens within few hours actually, and adaptive immunity takes place after 5 to 7 days. And so the innate immunity tries to tackle an infection. When it cannot, it calls on the adaptive immunity. So it is the adaptive immunity through which our, all the principle of vaccine is through our adaptive immunity. So this is a macrophage taking up the Streptococcus bacteria. And many years ago, we showed that these macrophages, when they take in, they actually generate reactive oxygen species within something called the phagolysosomes. So the bacteria or any of the pathogens come in, they are in a special vacuole within the cell. And then this cell sends its lysosomes. The lysosomes fuse with this particular phagosome. And then the phagolysosome, the bacteria or any pathogen is degraded. Some of it is presented to the surface to interact with, and many of them are eliminated for recycled as raw materials for the cell. So the reactive oxygen species that comes in here are mostly superoxide, which is formed by the NADPH oxidase located on the phagosomal membrane. And the superoxide generates hydrogen peroxide, which under superoxide dismutase actually forms the hydroxyl radicals, the peroxyl radicals, and eventually with nitric oxide, it forms the peroxynitride. All these very reactive molecules actually are helpful in degrading these along with the lytic enzymes that are present in the phagolysosomes. So this is how the antigen is tackled by innate immune cells in the first go. If they fail, because the bacteria, the virus, they have acquired many tools to actually thwart these actions by the host cells, and they can prevent the host cells from taking these actions, and then they can survive within the host cells. So now, if you look at the adaptive immune response, this is a dendritic cell, and it is talking to a T cell. But this talking is very complicated. If you look at this infographic, you will see that there are multiple molecules which helps presentation of the chewed up portion of the pathogen to the T cell, and through which is called a major histocompatibility complex. There are molecules over there. They present the antigen to the T cell, and the T cell receptor receives it. Then signaling happens within the T cells, and the T cells can form cytotoxic T cells, help in generation of cytotoxic T cells, and it does stimulate B cells, and the B cells form the blast. Then they form the plasma cells, which actually secrete the antibodies, and there is a population which goes as memory cells in our lymph nodes and is stored there. So in future, if there is an attack, these memory cells will be called out. So this is the principle on which the vaccine actually works. So here also you see there is a macrophage, which when it picks up the bacteria, these are all pseudo-colored scanning micrographs. When it picks up the bacteria, the streptococcus, it presents its antigen to the T cells, which is sitting on top. And then this actually goes and talks to the B cells or makes more cytotoxic T cells. And that is how our immune system works. So the T cell recognition, as I said, natural killer cells or cytotoxic T cells recognize and infected cells and kill them, so that the infected cells die, taking the virus with them. So some mutations in the coronavirus may enable the virus to avoid binding to antibodies and not recognize T cells. So there are mutations that have happened in the coronavirus that helps them to avoid these initial assaults by our own immune system. Like in spike proteins have 1 to 6 T cell epitopes. So if one of the T cell epitopes mutates, the infected cell will actually not be recognized by the killer T cells. So it is very crucial that surveillance of genetic and antigenic changes that is happening to the global virus population. And that is being done all over the world. We are actually sequencing the virus to see what are the genetic changes and antigenic changes are taking place and how they can actually avoid the host onslaught. So here is a cytotoxic T cell again, which is actually attacking another cell, which is a tumor cell in this case. But the cytotoxic T cells are the perforin granzyme system kill host cells which are infected. So this is just a summary of what I said, that when the SARS virus comes in, it enters into the cell through ACE2 receptor, and the viral RNA is translated into proteins. There is a virus assembly, and virus is again given out. But when in case of the response of the immune system, the viral peptide is presented by the dendritic cells or the macrophages to T helper cells, which generates cytotoxic T cells, which kills the infected cells, and the B cell generates the antibodies. So this is how our immune system plays a very vital role in case of natural immunity, in case of vaccines, and in case of when infections are taking place. So our immune system has to be very strong in order to interact in this way and save us. But as I said, our immune system gets less time to evolve itself than a virus or any pathogen, because we have less generation time. So with no effective treatment options currently, we have only the vaccines. So as I said, that immune system is required for the vaccines, required to thwart infections. Double-edged sword, this immune system can at times cause significant damage in the form of autoimmune reactions. And what we have heard about cytokine storm, here the immune system actually goes into overdrive and causes the cytokine storm. That is another handicap that we have with the immune system, is that when infection happens, there is an antiviral response and T cells are activated, as I said. But T cells which generate interferons that are most, they are the primary antiviral compounds, they can be inhibited by defective interferon production and presence of autoantibodies to interferons. So autoantibodies to interferons, again, is the handiwork of our own immune system, but defective in interferon production, maybe the virus is causing something. There are ways that the virus can disrupt interferon production or binding of interferon to its receptor. So it can also, it can generate decoy receptors and prevent binding of the interferons. And it perturbs also the intracellular signaling pathway and, directly, downregulating the level of expression of interferon stimulating genes. That is another way the virus can go. So, immune system actually can. Now, very recently, I think it was last week, that report came in that human immunodeficiency virus, which is extremely persistent and can lurk within the body for decades and attacks immune cells, and that compromises a person's ability to fight other infections. So the immune system is downregulated. And so they always progress into AIDS, but very recently, for the second time in the world, researchers have found a person whose body seems to have managed to rid itself of the virus. So are these ultra-powerful T cells? Are we capable of generating ultra-powerful T cells amongst ourselves within our body? This gives us hope that human immune system is powerful enough to control HIV and eliminate all the functioning virus. Then is it selection working? Or there are just two individuals that have happened into, but which shows us that there is a possibility that ultra-powerful T cells, we have the capability of doing that, only that we just don't know why it happened and how it happened. So this is why basic research is very important, and we need to understand these nuances. Now, before COVID-19 happened, actually, this is a report which came out in that there has been substantial reduction in deaths from infectious diseases in the high-income countries, particularly the United States, where the mortality fell from about 800 per 100,000 people, which had in 1900s, to 50 per 100,000 people in 1950. So from a 50% death to about 6% death it came. And over the past three decades, the same thing has happened in low and middle income countries as well, in Asia and in sub-Saharan Africa. As immunization coverage in children has increased. And so that deaths among children younger than 5 years also have decreased, and deaths in malaria have been decreased by using measures, although we don't have a malaria vaccine, by using measures which controls the vector and which actually causes less death to people getting affected with malaria. And HIV infection rate has also fallen thanks to all the drugs that has come out, because although we do not have a vaccine for HIV, we do have many drugs. So, and poliovirus also now stands on the brink of eradication. So we have been successful in doing so. There is hope that we can eliminate other diseases that keeps the emerging diseases that keeps on coming in. Now, this remarkable progress was because of the increased investment in healthcare infrastructure, better diagnosis, better development of vaccines, better distribution of safe and effective vaccines, increased acceptance by people. But this is a problem the world over, that people don't accept vaccines for various reasons. So in 2016, after the Ebola crisis, the Coalition of Epidemic Preparedness Innovations was created, which is CEPI. Now, CEPI was designed as a public-private partnership to accelerate the discovery and development of vaccines against known various pathogens. So CEPI supported the development of messenger RNA and DNA vaccines, and that is what we used from that platform for the COVID vaccines. So these kind of enterprises are necessary to create platforms which can be used in case of emergency. So after all this is a cover picture from the journal Emerging Infectious Diseases, and this drawing was by Hokusai, Katsushika Hokusai, who was a very famous Japanese painter. And he drew this giant wave where he has some fishermen here who are so helpless. So there was an article of tidal waves and human frailty by Potter, who is the author. Now, she was from the Center of Disease Control and Prevention. And this was a sort of that how helpless we are against this tidal wave. So we have been hit by the COVID-19 like a tsunami, but we have stood our ground. It's true that many deaths have occurred, but then we have also been able to understand the virus, to understand the disease, and come up very fast with vaccines. Drugs we have not come out with as vaccines we have done, but still there is hope, and we will fight these infections as they come in. And it is only and only through investments in basic sciences that we will understand, because all these sequencing that happened of the virus, and within 1 month, a whole lot of sequences got done from December 2019 to January 2020 when this happened. And it is because of science, because of basic science, that this was possible to have those sequences, have the immunogenic epitopes formulated. So I think we don't need to despair, really, but we all are part of this effort. And I think it's going to be a challenging time, because there are so many good papers which has come out, and we've been able to use the modern tools of biology to understand much of the disease. And now because of this emergency, there is a network all around the world to fight this disease, which is a very, very happy thing to happen. In the sense, in the backdrop of the tragedy of the coronavirus, this worldwide collaboration is very important. Otherwise, we will not be able to tackle a virus like this. So the future, as I said, science has discovered much about pathogen biology and made extraordinary strides on the development of vaccines and drugs. But funding needs to be improved for basic research. The future will depend on the ongoing evolution of the existing and emerging pathogens, the behavior of people, the decisions of the governments, and on the development of treatments and preventive vaccines. For this pandemic, unless high vaccine coverage is achieved, premature stoppage of mask wearing, hand washing, and social distancing would be catastrophic, that we are seeing in Europe right now. So we know so much from the early days of medicine, yet we do not. And certainly, as another artist puts it, that we don't want to be like this for the rest of our lives. Thank you very much.
Thank you. Thank you, Professor Shaha, for giving a very elaborative talk about the in-depth issues connected with this emerging disease and the complete immune system. I think there is no dimension which you have left, and you have covered from the immune system to what is the way forward, and how they erupted, how the different kind of viruses, and especially the coronavirus connected issues, which generally people have no knowledge so much. So there is one question, ma'am, if you allow.
Yes, please.
And that is what triggers autoimmune disease? How to control or manage them? It's a question by Dr. Hari Prasanna, our own institute, Indian Institute of Millet Research in Hyderabad.
Yes, as I said, that autoimmune diseases I mean, our immune system is the cause of autoimmune. Reason is there are perhaps by some genetic predisposition or the particular environment within the body, the immune cells react against the body itself. So the autoimmune diseases are a fallout of that. And we understand partly what autoimmune diseases are, and in many cases for treatment, these suppressants are used for the immune system, so that the autoimmune diseases, they come down. For example, one is a very common one is arthritis. That is an autoimmune disease where our joints get affected, and because of the immune cells, attacking our own cells. So this preventing these is very difficult, because this is something, because with some people, it will happen, for some people it will not happen. So it's very individual in that respect. So autoimmune diseases are very difficult things to tackle, and doesn't happen to everyone, because it's the decision of the immune system of the body.
Thank you, ma'am. There is another question by Nilima Garg. What causes cancer of lymphocytes?
Well, it is probably a mutation that happens in the that is in the bone marrow cells where they are coming from, and these are the mutations that cause the excess proliferation of the cell, and that is what is responsible for the lymphoma that we have.
Thank you. So there is no other query or question, and I'm happy to inform that our Secretary DARE and DG ICAR, Dr. Mahapatraji, he joined just after 5 minutes, and he listened to your lecture. So I request him, he was chairing this session, to give his remarks.
Okay. Just going to be afternoon, but very good morning to everybody. And I, at the outset, let me express our sincere gratitude to you, Dr. Chandrima Shaha, Madam, for kindly agreeing to our request and to be part of this lecture series, that 75 lectures as a part of Azadi ka Amrit Mahotsav. And we have been actually listening to many learned experts on various topics. And today, you have taken us minds, and it was so mind-blowing and fantastic. And your deep understanding in the subject was very much evident. And also the kind of the way you were explaining, and a wonderful teacher that you are in explaining very complex things in a very simple way. And that was really very, very rewarding and enriching for all of us. So we would like to give you a big hand. We can't have a standing ovation and all online mode.
Thank you.
So thank you very much. And, we really appreciate not only to kindly agreeing, but time is over. So I don't want to really take more time. But a few words I would, certainly would like to use to describe and summarize very briefly. And so this world of human diseases, as you started with fundamentals, the pathogenicity, establishing with those fundamental experiments, and whether it is Robert Koch's experiment, and even Darwin's own theory, and the way you have explained those fundamental concepts, I believe my colleagues, they would have, they were all taken back to their, those days of learnings, basics, and understanding in simpler terms. The way you explained those was so well explained, madam. So thank you very much, and we really understood and gained those insights. But more important is that how these RNA viruses are actually evolving, and the process, creating havoc. And you cited several examples, the flu, for instance, and the earlier flu and today's flu, and then flu continues in various forms, and then putting all of us in deep trouble. And the kind of mutation rates that you described, that 2 mutations per month, that's what you highlighted. And that's something which is very, very significant. We often thought that viruses are inert, non-living, and very difficult to describe them if else. And the other thing is standstill, nothing happens. And once they enter the living system, and then they create havoc by way of multiplying and utilizing the host machinery. And more importantly, the kind of capacity which increases because of the mutation. For instance, as you said, that mutation are 226 position in E1 protein, providing enhanced vectorial capacity for the virus. And that's something which is suggesting and revealing that how those mutations are bringing in greater adaptability and infectivity, virulence in these so-called inert one, but in the host system it is doing everything, mutating itself, and then enhancing its adaptability, and then creating havoc. So I think those points which you have made, and in fact informed us, all of us, those who are not working in this area, have in fact understood this very fundamental, that why in the itself is also RNA virus, and why things are so difficult to manage, and frequent mutations making it far more challenging. You also highlighted various other things, and very briefly to summarize the kind of evolutionary rescue that you highlighted. Again, a kind of learning experience for us, that obviously viruses, or for that matter, any infectious organism is becoming extinct because of lack of evolution happening, and mutations not really supporting this. And it's getting eliminated in the process. It takes time to really have that, and that's what you have highlighted while describing evolutionary rescue, and informing all of us that how viruses or organisms wait for that kind of mutation to arise, and in the process, again, creating, the widespread, infection and disease in the process. You did mention about the evolutionary trade-off, and that was another important concept for us to take, back home. That how milder infections are happening, because once the organism, the host goes, obviously the virus goes in the process. Would like to persist, and the process you have highlighted how milder infections are happening, long persistence is happening, and carried forward a very longer period of time. In that context is obviously HIV, 40 years, we don't have vaccines, and they are persisting there, and that ultimately expressing in the form of AIDS. So that's beautiful examples of milder situations, and then ultimately transforming into serious challenges at some point. So the kind of immunity system that you have explained very lucidly, whether it is innate immunity or adaptive kind of immunity system. In plants, we don't have that kind of immune system, but innate system operating very well. And we have the expression of SOD, for instance, and certainly the kind of SOD or catalase and other enzymes that you have highlighted, and how the innate mechanism operates, and how it really helps to protect, and very well described and informed us. It's quite educative. And similarly, the adaptive mechanism in the context of that you highlighted, so important to save, and that's how the population, our actually survival rate has increased over years, and that's what you have highlighted. And in 1900, how people were dying, 800 per a lakh population to now 50. So obviously, the vaccine has played a very significant role, in addition to other drugs and antibiotics and so on and so forth. So all that contributing so significantly. So in the process, what you highlighted, not only the fundamental, how the T helper cells leading to, for instance, cytotoxic T cell or B cells, and B cells again providing us antibodies, and and then T cell cytotoxic ones or antibodies fighting the antigens through the mechanisms that you so nicely described. But the way you described the mutation rate in spike protein of the coronavirus, and particularly one or two mutations in these kind of regions which are actually T cell epitope specific. That's the reason why it has been quite challenging and escaping the recognition by T cells. So that's a very interesting one, and we really appreciate your very, very deeper understanding, first of all, and also explaining in illustrative terms how really this is happening. But then the need of understanding in this context the challenges that before us that we need to really address. We first of all, as you highlighted, that basic science is so important, and we need to really do and keep doing basic science of all these emerging and challenging diseases, so that we have a quicker response, as in case of corona you highlighted. And that's what is the important lesson that we can get from your lecture. We need to have those fundamental sciences done, and so that we work out quickly the solutions to address these challenges, so that in the human population or even animal population, we suffer. This needs networking, and as you rightly pointed out, the global networking, and also we must capitalize on this strength and understanding. The new pathogens, emerging diseases should be very vital, and the expertise is distributed across the world, and there are different capacities. And we need to really take advantage of these through collaborative efforts. That's what you highlighted. And obviously, the need to understand evolution. And that's what you have highlighted, the fundamentals. And we need to really understand how evolution is happening, whether it is in the spike protein or other regions of virulence and pathogenicity and all that. And so they would be quite helpful in designing drugs and targeting specifically the drugs to the regions of interest. And you also highlighted how public-private partnership has emerged as a important strategy to fight these diseases. There has to be greater investment, and private obviously gains from this by way of vaccines and drugs, and their engagement from the beginning would be so vital to fight these emerging diseases. So I think that's another important point you have highlighted. But then the government policy and response, quick response, and that's what happened in India. And you also highlighted, unless we have greater investment, unless we have right kind of policy and decision making happening at a fast pace by the government, obviously we will be defeated. So that is another area you have highlighted. And I believe not only in this case, more so in such situations where the faster mutation rate happening and evolution happening and adaptation happening. And obviously we have to have very fast response in terms of policy, decision-making, investment. Otherwise we will be again defeated and eliminated faster than before. And obviously at the end you did mention that we have to really accept these technologies. Unless accept, obviously we'll be defeated. Acceptance of action, has not really happened in countries like USA. There are people who have not really accepted vaccine so far. So there are regions, and there are, in case of plants, GM, gene editing. So acceptance has not really happened. So I think there has to be massive awareness and public education of science. And so that's what our academies, our people and scientists have to really work in that direction. Need to really make people aware of the power of technology, the safety of the technology in human and environment, and adequate going to those end users so that they are educated and informed. Because unless they're informed, probably we will not be able to save. Because if there are carriers who are not really taking the vaccines and drugs, so they would be infecting subsequently. So we can't really eradicate anything in true sense if it doesn't really happen that way. And polio is example in recent history how in an animal system, our vaccine system is quite old. We have developed 25 vaccines, and starting from beginning of the last century, we started developing vaccines for animal system and eradicated diseases like rinderpest. And we continue developing vaccines. We have vast experience in this area. Our animal scientists have been working on this. Indian Veterinary Research Institute has been working on this. So I think acceptance of these new technologies is very, very important. And we should not forget that some of these 60%, more than 60% are zoonotic in nature. So unless we actually address in animals, obviously we will not be able to address in human. So in the context of One Health, this also assumes greater significance. We need to really build that kind of thing. And we have been trying to find out vaccine for animal. So corona vaccine for animal. And we are also similarly developed one and is under testing similar to Covaxin. And we hopefully, we'll be able to really deliver after testing. We're testing in dogs and other systems, animal models. So I believe that this whole world is so rich, and we have so much opportunity actually. And your lecture today is so revealing that how we can really have cross learning and then understand deeper. And people like you with so much of vast understanding in this area is actually a eye-opener for all of us. And we express our sincere thanks and gratitude to you for agreeing to our request and delivering such a wonderful talk. And at the end, I once again thank you profusely for this wonderful talk today. And our lecture series is really successful with your, you being here today with us. So thank you, Dr. Agrawal, for organizing this whole series and also arranging this lecture today. So thanks to all colleagues who are linked today to listen to Dr. Chandrima Shaha and his brilliant lecture. So thank you very much, Dr. Chandrima Shaha, madam. Thank you. Namaskar.
Thank you, sir. Thank you very much for nicely summarizing and giving the account of the ICAR's activities in the veterinary science and the other things which we have taken as a lead institute in India for the One Health. And thank you, Professor Shaha, again for this wonderful talk and for a very single request. You accepted our invitation to deliver this lecture. So really, I am so thankful to you for this wonderful lecture. And thanks to all our colleagues who have joined today from universities, from our ICAR institutes, the senior faculty, and other scientists who are involved in this. And thanks to one and all who are connected through the live streaming, who are connected on the other platform. So thank you all very much. And please stay connected for this important series, which we are conducting every lecture 1 week, one lecture 1 week. So in that, sir, I'm happy to inform you that now we are almost halfway through. We have to conduct 75 lectures, and today is the 36th lecture. So almost we are in midway.
Okay. So really we finish this with a big thank to Dr. Thank you, madam.
Thank you.
Namaste.